The following video is a recording of a lecture from Genes to Galaxies, the 35th Professor Harry Messel International Science School, presented to high-achieving Year 11 and 12 students from across Australia and 10 other countries.
Okay, so thank you Adam. It's nice to hear my bio. So what I want to talk to you today about is asthma and airway remodeling. And as Adam's introduced, I'm a medical researcher. So I think you've heard a lot from other fields, and you've also had a good introduction and in-depth look at genes as well. What I want to talk about is how those genes are important in disease. And the particular diseases I'm going to focus on today are asthma, and which I know you know some about, and we're going to overview some of the knowledge in those areas. And also another disease you might not know that much about, which is called airway remodeling. And I'm in the Faculty of Pharmacy, and I teach to pharmacy students about drugs and about treatments. And so the research that I have going on in my lab is about understanding how the drugs that we currently have work and what could we do to improve those? So it's from a medical research, but towards a treatment point of view that I'm going to be talking. Okay, so let me bring some slides up and we'll do some information first. So let's think about this concept. Now, asthma, as we know it, and you know it, is a disease that causes bronchoconstriction. We'll look at the cellular mechanism of that. But just have a think about breathing right now. This slogan, "When you can't breathe, nothing else matters," is the slogan of the American Lung Association. So breathe, just breathe at the moment. So breathing is something you do involuntary. You just do it. You just don't really think about it anymore. It's one of those involuntary actions that you do. It's part of us. If we can't breathe, well then nothing else matters. If there's other diseases, cancer, whatever, you can't breathe, nothing else matters. So breath is something that's so important to us. But what actually happens in asthma and in the other disease that I'm going to talk about is that in asthma, we have an airway flow limitation. So it is difficult for asthmatics to breathe without the drugs that have been developed through research. And airway remodeling is an area that we're going to get into, which is another area where we need a lot more work. And in the final part of my talk, I'm going to be talking about the research that we have going on in my lab, and we're going to get down to the cellular level. We're going to talk about a family of kinases, enzymes, called mitogen-activated protein kinases, and they're the possibility for future therapeutics. This may be a new drug target in a couple of years, and that's what I'm going to be talking about. So first off, let's refresh our memories about asthma and its importance in breathing. So, where's the best place for videos? YouTube?
Through the nose and mouth enters the respiratory passages. Then, the air entering the respiratory system passes through the trachea, then splits into the bronchi, bronchioles, and finally enters tiny sacs called alveoli, where the exchange of gases takes place. Asthma is a disorder of obstruction to breathing due to inflammation and narrowing of the bronchial tubes. Initially, the bronchial tubes become inflamed and produce thick mucus. Later, the muscles surrounding these airways tighten, and air cannot move freely. This is called bronchospasm. The result is shortness of breath, and the air moving through the tightened airways causes a whistling sound known as wheezing. Pollen, pet dander, weather changes, tobacco smoke, etc. can trigger and worsen asthma symptoms in susceptible patients. Short-acting bronchodilator drugs, which can be inhaled, provide immediate dilation of the constricted bronchi.
Okay, so we're going to talk about some of those aspects. So that was just an overview. I'm sure you know what asthma is about, but when you breathe, the airway in an asthmatic becomes tight. So we're talking tubes here, where airways can really be broken down into a series of tubes through which the air flows to the alveoli. So as we heard in that video, asthma, when you work in America, as I did, I have to go asthma, is a disease where the airway tightens. There was a couple of different things in that video that we're going to focus in on today. Inflammation in particular. Inflammation, you would know, is an important part of health and disease. When you cut yourself, it's an inflammatory reaction that allows wound healing. But inflammation in diseases such as asthma is one of the reasons why that tightening of the airway occurs. So inflammatory mechanisms offer a lot of promise in terms of drug discovery. And one at the end of that video, they talked about puffer, about a bronchodilator. And we'll see how that can actually reverse the symptoms of asthma. So, thinking of asthma and its relevance in Australia. It's a very important disease. As Adam mentioned, you may be affected by asthma or you know someone who is. In Australia, the prevalence of asthma is quite high, around 10%. So it's a very important disease from a socio-economic point of view. So around 2 million people in Australia, 1 in 4 children and 1 in 7 adolescents are affected by asthma. It's a very widespread chronic health problem. Now, I put this slide in as well because I know we have international students here as well. So here is a graph that shows how the prevalence of asthma on the x-axis here, and on the y-axis, different countries. So you can see at the top, and UK is at the top of this of the scale, not a very great thing to be proud of, but I know we've got some people from the UK here, has the highest prevalence. Here's Australia, New Zealand, and really as a general rule you would say the more Western countries have a higher prevalence of asthma, and we can talk about that a little bit more in questions. But there is an increasing prevalence of asthma, and it is something that we hear a lot about. So asthma, it is something where the airways are narrowing. Let's just think about it, and we need to get down to a cellular level and then a genetic level in terms of what is actually happening. So as was mentioned on that YouTube video, there's a number of different triggers for asthma. People have a variety of different things that will cause an asthmatic reaction. And in this example and in the chapter, I wrote about allergic asthma. This is not the only type, but this is a way in which asthma is triggered. So people can be allergic to certain things, certain triggers such as exercise, allergies, cold temperatures, infections. But if you are allergic to particular things, you will have a sensitivity to those things. So here, it just came into this slide, is a creature. Does anyone know what that is? Dust mite. So Dermatophycoides tenuis is its full name. And why is it allergenic? Why does it cause allergy? It's around the picture there. Feces, that's it. It's not that house dust mite live in your beds. They eat all your dead skin. They eat your hair. It's really quite gross, actually. And it is not the house dust mite that people are allergic to. It's the feces, the proteins in the feces of the house dust mite. So the feces, and they see the little feces there in that picture, have proteins that if you're allergic to house dust mite, you will react to. You have antibodies towards those proteins. And house dust mite allergy is very high in Australia. It's not as high in other countries. When I was working in America, as Adam said, that predominant allergen was cockroach. So if you would ask, well, why cockroaches rather than house dust mite, there was a lot more cockroaches rather than house dust mite. It's around the moisture that's in the house. But if you analyse the feces of house dust mite, compared to cockroaches, you would see the same sort of proteins. So there were proteins that people are sensitised to. So if you are unlucky enough to be, have an allergy to house dust mite, you're in bed, you're rolled over, and you rustled up some of the house dust mite feces that you live with, you would breathe in allergens, and then a series of chemical cell biological reactions would occur. The first is the mast cell. So you have breathed in the allergen. The mast cells are in your airway, so now you have got cells interacting with those proteins. The mast cells are the first inflammatory cell to see the allergen. It has on its surface antibodies to that allergen. When the allergen cross-reacts those antibodies, that whole mast cell will explode, as was shown here in this cartoon. The allergens attach and the cell degranulates. All of the proteins that are in that cell now are exposed and are in the inflammatory milieu in the lung. And what will happen is a series of cell biological interactions. Cells in the lung will cells from the blood will be recruited into the lung. So through your lungs, you've got a lot of vessels. The chemical messengers that have gone from the mast cell will cause the other cells in the blood vessels to come through. So shown here in this slide, here's a blood vessel passing through the lung. It senses the messages that the mast cell has given and comes into the lung as well. So we're starting to get a really inflamed lung. Further cells will be induced to come in, and further proteins will be produced. We'll come back to this, but of course when you have cells making proteins, this is all a response of genes being transcribed. So you know a lot about genes, and genes are being transcribed here, and proteins are being made in these cells. This gives us a clue as to how we can therapeutically target them. These molecules released from these inflammatory cells will act on receptors to cause what we have just seen in that video as a bronchoconstriction. The molecules, chemicals, will interact with specific receptors and cause bronchoconstriction. So what that is, now you've got a lung that's full of inflammatory mediators, chemicals floating around, interacting with specific receptors. There is a specific receptor on airway smooth muscle cells that are, if you consider the airway as a tube, it has a ring of airway smooth muscle that contracts when it sees these mediators. It contracts, and if it was a tube, it's going to be narrower. You guys know that if you have a tube of a narrower diameter, it is harder for the air to go through. So bronchospasm, as we know it as asthma, is all mediated by a cell biological interaction where a mediator has induced a contraction. So we know this, we've known this for many years. What do we do to tackle it? What sort of drugs are out there? How do you inhibit bronchospasm, so the bronchoconstriction? Does anyone know the names of any of the drugs that you use to do that? Sorry, you said? Ventolin, yep. Corticosteroids, yeah. Ventolin, yep. Another one? Symbicort, yep. Propanol. So there's a lot of different drugs that will have an effect on this. So some of the ones that were called out were, there's two different places where there's control of asthma. The first group of drugs are called beta-2 agonists, and we're not going to get into why the receptor mechanisms behind that. But we had Ventolin and we had various other beta agonists that alleviate that bronchospasm. So if the airway contracts when it sees the house dust mite, if you take a reliever, such as Ventolin that was called out, that's going to relax that muscle. So the airway will relax and no longer be as tight as it was. The other one couple that were called out were steroids, corticosteroids. So corticosteroids are what are known as preventers, and they prevent that inflammation. So in terms of asthma therapy and acute asthma, in an asthma attack as we know it, we have very good drugs to be able to tackle them. The relievers, shown here, and the preventers, which are the corticosteroids. They will reverse this bronchospasm and very effectively have symptom control of asthma. So it's very fair to say that asthma is a very treatable health condition. Through years of research, probably about 4 decades worth of research now, all the biochemical pathways have been understood in acute asthma attack, and we have a large number of drugs that can effectively treat them. So that's very, very good news. So asthma management has allowed us to tackle acute asthma attacks. And because of good asthma management, asthmatics, as you know, and some of you may be asthmatics, lead normal, healthy lives. It is a condition with appropriate treatment, appropriate management. It's really a non-event. It can be. Here are some famous asthmatics, and those who are in asthmatics would know that these guys are the poster children for, and poster women. Pink, Lindsay Lohan, Grant Hackett, and Elizabeth Taylor, all famous asthmatics. You might have some of the people and you guys might be some other famous sports stars and celebrities with asthma as well. So, asthma management and the knowledge of asthma and the drugs that have been developed are very, very useful and effective for living with bronchospasm. Let's get to the unanswered questions. The unanswered questions around asthma and the consequence of asthma, which is airway remodeling. Has anyone heard of this concept before? Airway remodeling. Okay, so what sort of stuff do you know about it? Chronic, yep. Very good. So the chronic condition, more cells growing and scar tissue is a consequence of this asthma and inflammation that we see. And it's something that we need more work on, and something that I work on in my lab. So, what is it and how can we treat it? So, looking at our lung again as a tube, here is our normal lung, normal bronchus. This is the lumen, the hole in the middle where the air goes through. These are the epithelial cells that are surrounded. This is this airway smooth muscle that I was talking about, the muscle that contracts and is responsible for the tube tightening. So this is a normal airway. And here, if we compare it to an airway that has been remodeled, it has a narrower lumen. We have mucus in the lumen. We have some hyperplasia of the cells, so that means more cells, more epithelial cells that surround the lumen. And in this case, we've also got more airway smooth muscle. So your colleague here said that it's like more cells, more scar tissue. This is what is considered. Also, there's a greater amount of fibrosis. So fibrosis is what is sometimes known as scar tissue. So we have a number of extra proteins growing here. We have fibrotic proteins, collagens. We have a number of cells, increased number of airway smooth muscle cells. Also a result of gene transcription and protein production. We have hyperplasia, increased number of epithelial cells, more genes, more production of protein products. And why does this occur? It is considered that if asthma treatment with the drugs that we've just talked about, the beta agonists and the corticosteroids, is not effective or is not managed well, that as a consequence of that ongoing inflammation, that there will be some structural remodeling of the cell. The cell will start to become somewhat scarred, if you will. It will start to change shape and it will become what is sometimes referred to as thickened. It has been modelled that the airway is much smaller in the lumen, and of course then if it's got more airway smooth muscle in it, in response to the same house dust mite allergen, it will contract and also be able to reduce the lumen size. So there's more muscle, a smaller lumen diameter, so in response to a contractile agonist, the airway will tighten even more. So there will be a decline over time of lung function. So this is some concept that has now been raised after knowledge of asthma and the inflammatory aspects of asthma. We now know over time, that the airways of asthmatics become thickened or remodeled. And this is really because the inflammation that we've seen as part of that asthmatic acute response, all those cells flooding into your airway, all those cells flooding into your lung, if it's not controlled or treated properly with the corticosteroids and the beta agonists, remodeling will happen. The cells will respond to all these exogenous stimuli that are out there and will start to form this scar tissue. This is what it looks like in some human samples. These are cross sections of lungs from a 13-year-old child who had died from in a car accident, so it was a non-asthmatic situation. And this is a cross section of an asthmatic airway from an age-matched person who had died from an asthmatic attack. And of course, at this stage, I'd like to acknowledge the parents of these children who had donated these lungs for research purposes. So let's look at the differences here. So you've seen it in the schematic. You've seen that this is airway smooth muscle here. This is how much you normally get in a normal, a non-asthmatic airway. And here it is much, much thickened. We've got a lot more of it in an asthmatic airway. So the cells have grown, basically. Got a lot more mucus in our asthmatic airway as opposed to our non-asthmatic, and there's various other contractile modifications. There's an increased basement membrane and an overall narrowing of the lumen. So this is what we're trying to work on now. What is this remodeling? How can we treat it? There are some challenges out there, and there's some possibility for some future therapeutics. What we know is that it is because of uncontrolled asthma and uncontrolled inflammation. Now, unfortunately, they can't be permanent. We currently don't have anything to totally reverse that phenotype. We want to make the airway back to what it used to be. That's an issue that we're trying to work on now. Also, as development of the remodeled airways is correlated with the deterioration of lung function, really there needs to be a further look into therapies that can reduce those structural changes. Corticosteroids, they can inhibit some aspects of this remodeling that we've seen, but not all, and also we have to use corticosteroids very well because there are some side effects, particularly in children. So there's always a look out there in asthma research for what's called corticosteroid sparing. So therapies where you can reduce the amount of corticosteroid and have an effect on airway remodeling. So what sort of things can we do? From my point of view, what we've been looking at, there are a number of different things out there. So in medical research, what happens around the world is that people research different areas and develop different niches that they're interested in. I am going to be talking to you in the second bit of this lecture about the mitogen activated protein kinases because I feel it is an important place for us to look further, do more research as a possible therapeutic target and ways in which to inhibit this airway remodeling. Many other people around the world are doing other things. What happens is that various labs work on different things, and together we'll be able to answer this question. It could be mitogen activated protein kinases, and I hope it will be, because that's what I think it is, and I'm working on quite hard, but other people may think it's other aspects. So this is a healthy exchange of research that you know about, and you would have seen in some of the lectures that have been given, that there's always people competing across the world. We come together, we give presentations at scientific conferences internationally, we exchange ideas and hopefully come up with an answer that will help human health. So where I want to go to now is to talk about mitogen activated protein kinases, why they're important and what I think we can do. So, where we're at now, and let's put it into context of what you already know. If this is our normal airway, and this is our airway that's become remodeled, increased airway smooth muscle, decreased diameter, increased mucus, more blood vessels, scar tissue, how can we stop going from here to here? What we know is, through about a decade's worth of research, just summarised on one slide, is that underlying airway inflammation and remodelling is the synthesis of a number of different molecules. So the synthesis of pro-inflammatory molecules, such as the cytokines, chemokines, lipid mediators, growth factors and enzymes. So the sort of things in our animation where we saw all those cells flood in to the airway. What some of those, what they are called cytokines. So cells talk to each other, basically. Then we are all a mass of cells, but the only way your cells can talk to each other is because there's signalling between them. In this slide, we see that there are things called cytokines. What cytokines do, one cell produces a cytokine. It produces it after there's gene transcription and produces a protein. It is secreted from the cell and it will tell another cell, you need to come in here. So what we saw in the lung example was that the eosinophils produced cytokines. They then told other cells, come to this place. This is where the party's at. This is the inflammation centre. These are all proteins. Proteins are all a response of gene transcription. So on this slide, we see that what underlies inflammation and remodelling are the production of a number of different inflammatory molecules. Cytokines, chemokines, chemotactic cytokines as well, lipid mediators, so molecules that are made up of fats, growth factors and enzymes, all the product of gene transcription and from DNA. What is pivotal in all of this is after inflammatory stimuli, there is one pathway that underlies the production of all of these different genes, and it's the MAP kinases. And we're going to hear about what they are in a second. So when you get inflammatory stimuli, you get this signalling pathway called MAP kinases activated, and the regulation of inflammatory genes activated. It will then be responsible for producing all of these things that cause inflammation in the lung, and through that inflammation in the lung, we have this remodelling. So, the question is now, what are MAP kinases? Well, obviously, since they're in this talk, they are a signalling pathway that underlie what we've seen. We go from the house dust mite to the cytokine secretion to bronchospasm, predominantly via MAP kinase pathways. And MAP kinase pathways, what are they? They are chemical cellular signalling molecules. So when you have a cell, your cell has to talk from the outside to the inside. It has to receive information from the outside and turn that information into something. So here is a schematic a very general schematic of cellular signaling. And this is what's happening within your cell. So if you can imagine, this is the this would be, follow my cursor, that would be the outside of the cell. Here is an extracellular signaling molecule, something released from histamine that's going to cause a contraction of a cell. It's floating around in the lung and it interacts with a specific receptor. There are a number of then signaling proteins that are turned on to pass that message on. So signaling is sometimes called signal transduction. It's a relay where information is passed from here to here to here to change gene expression. So you've heard a lot about genes. These are the genes down here. Transcription occurs, but there are ways in which that message is turned on. And the message is relayed, if you will, like in a running race, by the molecules known as MAP kinases, mitogen activated protein kinases. So they are intracellular signaling molecules. What they do is once a molecule binds to a specific receptor, it is turned on. The signaling proteins pass on that message via phosphorylation. So it's an enzyme reaction where there's one small change, just one phosphorylation, that will then say, right, I'm turned on now, I will pass that message down to the gene. So mitogen activated protein kinases, abbreviated as MAP kinases, are signaling molecules that drive development of airway remodeling. So, like all cell signaling, there are many members of pathways, and there are 3 family members in this one. I've just put them up here: P38 MAP kinase, JNK, and ERK. In cell signaling, all we ever do is talk in three-letter abbreviations. Everything has to be very short, and that's how we talk. So P38, JNK, and ERK. The P in the first one stands for the size of the protein. It's 38 kilodalton. JNK was originally discovered by someone who thought it was just junk, but it wasn't. It was actually a very important molecule, and now it has the full name of C-Jun N-terminal kinase. ERK is just called ERK because it started off by extracellular signal-related kinase. So they originally have names that may make sense, or maybe we just get used to them as abbreviations. So what these things do, they're all enzymes, 3 enzymes. They are turned on when they're phosphorylated. They pass the message down to the gene. The gene says, okay, make some fibrotic proteins. Activation of MAP kinases requires phosphorylation. So let's just look at that in a brief animation before I get into how we're going to target them with drugs. And what the animation will show is a whole cell and how the cell then takes the signal from the outside of the cell into the gene. And in the pathway somewhere will be our MAP kinases, and I'll point it out. Okay, here's our cell. Here's the nucleus. Here's the gene promoter. And when I turn this on, we're going to see some receptors come together and a number of signaling molecules pass on a message via phosphorylation. Okay, so for phosphorylation, P for phosphorylation, that's now active. A number of signaling molecules are involved. Here's the first of our MAP kinases. Gets phosphorylated. Here's the next one. Passes on that message. Passes on that phosphorylation. Okay, there's ERK. This is our MAP kinase, one of our family members. I said P38, JNK, and ERK. Here's ERK. It's now activated. It has had, through seeing something from the outside of the cell being passed on to the inside of the cell, being phosphorylated, and now it's ready to cause gene transcription. These are transcription factors. Bind to the promoter of the gene and makes mRNA that will become proteins. And it will continue to make those proteins until we stop it. So that's a key bit. We'll come back to that in a couple of slides. So ERK is a MAP kinase. It's very responsible for this gene to be turned on. And so it moves into the nucleus. And if it didn't do that, hopefully you can see that the gene would not be turned on. So let me just go back to the slides. So that's where MAP kinase is involved in gene transcription. So what can we do with that knowledge? What should we do? If I said MAP kinases are so important in asthma and airway remodeling, which is true, and you've now seen where one of the MAP kinases is responsible for gene transcription and responsible then for cytokines, all those fibrotic proteins. And if it didn't move into the nucleus and start that gene transcription, well, then you wouldn't have that protein being produced. So obviously, then we can see that targeting mitogen-activated protein kinases are a possibility as future therapeutics in airway remodeling. So I think I've just said that with that. And there are two possible approaches. So in drug discovery today, the first approach is a very popular one. And it's not the one I'm taking, but it's the first approach. Small molecules, small chemical entities that will be able to get into the cell quite quickly and easily to inhibit enzymes are very, very popular. And we know a number of very good drugs that have been based on that. Gleevec in cancer therapy are small molecule inhibitors. So we can use small molecule inhibitors to get into a kinase pocket. Enzymes have pockets, and you can affect their activity by making molecules that fit into that pocket and block its activity. So the second one is to harness the power of an endogenous deactivator. Harnessing the power we have within our cells, they're very clever. Cells are very clever. They don't normally want you to do They don't want it normally be turned on all the time. They don't want to make fibrotic proteins all the time. They need to have some signals that turn them off again. And when you know cell biology, you'll see that there are kinases which phosphorylate things, and there are phosphatases that dephosphorylate things. So the kinases switch things on, and the phosphatases switch things off. So the cell has one of those MAP kinases, and it's known as MKP1. Mitogen activated protein kinase phosphatase 1, and it turns off MAP kinases. So we're going to talk about that in the final bit of the lecture. So first, small molecule inhibitors. I'm not going to go there very much in this section of the lecture. There are a large number of pharmaceutical companies making small molecules to target each of those three members of the MAP kinase family, P38, JNK, and ERK. Their code names are up there. They are molecules that were being used in vitro, so in the test tube at the moment. So we do experiments with airway cells from humans, and we have shown that when you use those small molecule inhibitors, they effectively inhibit the MAP kinase pathways and inhibit those functions that we know are important in remodeling. Those ones are being followed up by commercial companies, and will probably, P38 I think is the most advanced, and will probably be in the market earlier than the others. The final section I wanted to talk about would be what I am working on, what my team is working on. So here is an airway cell. I told you that when you get stimulation, you get MAP kinases. So any external stimulation there will activate MAP kinases, any of those family members. They will cause gene transcription of the proteins that gene transcription and production of proteins that control these aspects that are important in airway remodeling. So what I would like to do would be to increase the amount of an endogenous MAP kinase deactivator. So this MAP kinase deactivator, MKP1, it inhibits this pathway. So rather than interacting with the enzyme itself, what it does is dephosphorylate the MAP kinases. So if this gets turned on, MKP1 will turned on by phosphorylation, MKP1 will turn it off by taking off that phosphate group. It's a phosphatase. Takes off the phosphate group. Now this is always in our cells. It is an endogenous, so it is within our cells all the time. It's a MAP kinase deactivator. So that's what I want to do. I want to increase, my aim in my lab with my team is to increase the endogenous MAP kinase deactivators such as MKP1, to reduce MAP kinases, to inhibit airway remodeling. So, you know, that's fine, but how am I going to do that? That's the big question, and that's what we're currently working on. Now, how can we do this? How you can do these things, if you're a cell biologist like me, how you do these things is you learn a lot about the particular protein. So MKP1, I know now how you can increase it in a cell, and I can do that just in airway cells in the lab, and we do that all the time. We increase it, if our aim was to increase the amount of this protein, you can do it in 3 ways. First way you can increase is transcription. So you can increase the amount of mRNA for MKP1 and increase protein. The second way is you increase its post-transcription. And what that means is that once you've got your mRNA made, there are mechanisms within the cell that control how long that mRNA stays around. This is called post-transcriptional regulation. So you can understand that and try to interfere with it so that you can increase the post-transcriptional regulation of MKP1. So you get gene transcription, you've got mRNA that stays around a long time, and therefore, once transcribed, you'd have more protein, in theory. The third way is to look at its post-translational modification. So what is this? This is what I'm going to talk about just in this final bit of the lecture. Post-translational modification. I suppose I'm going to talk about it first because it's the first thing we're nearly finished on. And in labs, we like to do things that are pretty straightforward first so that we get some success, because otherwise it's research you probably heard from other lectures is a hard slog. You've got to keep yourself entertained with it. You've got to get passionate about what you're asking so that you can keep asking those questions every day. So we started with post-translational modifications because there was some, we felt that we're going to get somewhere pretty quickly. Let me tell you about post-translational modifications. There is a couple of different ways in which this occurs, but basically, has anyone heard of the proteasome? Proteasome, what does a proteasome do?
Breaks down proteins.
Yep. So I like the proteasome because it breaks down proteins, and the proteasome could be considered the garbage bin of the cell. And you need a proteasome, obviously, because you don't want to have proteins that have been made and their use has expired and they're just hanging around, because we'll just be a massive protein. So the proteasome is a very clever molecule complex. It's a whole lot of proteins together that can be considered the garbage bin of the cell. So MKP1, like a number of proteins, is inhibited, is broken down. Once it's made, it is very rapidly broken down by the proteasome. It is a protein that is made very quickly in response to stimulation. It turns off MAP kinases, and then it's not required anymore, so it is broken down. So our aim is to interfere with that degradation, and what we've done is we have a series of experiments going on now, and this is the only bit of data I'm going to show you, is to inhibit the proteasome, the garbage bin of the cell. So how you do that, there are a number of different inhibitors of the proteasome around, and the one that we've used, just for this figure, is called MG132, and it has the ability to inhibit the function of the proteasome. Now, in terms of MKP1, this is the Western blot. Does anyone know what Western blots? You know Western blots as well? You're a cell biologist extraordinaire. So Western blots are, you want to tell us in summary what a Western blot is? Yeah.
You remember the DNA gel that we did? Instead of agar, you take nitrocellulose and you run the protein down it.
Yep.
You blot it with antibodies.
Beautiful. Yep, and you buy these antibodies, and that is one from MKP1. Okay, so exactly right. What you do is that when you have a complex protein mixture, you run it down, you separate the proteins based on size, you transfer it to nitrocellulose, as you've said, and you blot with specific antibodies for a specific protein. So what this is are Western blots, as described. And here we have the top one where we haven't inhibited the proteasome. I've stimulated this cell, and we see that we have protein, our MKP1 protein, comes up at about 2 hours, and then it is degraded. So it peaks, and then it's not needed anymore, so it's degraded by the proteasome, the garbage bin of the cell. Here, in this case, we've added MG132. It inhibits the proteasome, and we're able to sustain our MKP1. We've got really robust upregulation of MKP1 protein. And now, by inhibiting the proteasome, we've been able to increase the amount of this endogenous deactivator of MAP kinases. And in the experiments that we just finished this week, we've shown that the opposite is true now, that we've got inhibition of the MAP kinases. If I showed you more Western blots, there'd be inhibition of the MAP kinases here. So, inhibiting the proteasome, the garbage bin of the cell, allows us to interfere with the post-translational modification of MKP1. So our aim to increase MKP1 via post-translational modification is what we're currently working on. And we have other stuff going on in the lab that I won't go into today, but I'm happy to talk to you about more, to look at the transcription and also the post-transcriptional regulation of MKP1. So what I wanted to just mention about inhibiting the proteasome, you can inhibit the garbage bin of the cell, that's fine, but that's going to inhibit the proteasomal degradation of all proteins. What we're currently working on is how to make that specific for MKP1. And we have a few really good leads to be able to do that. So what I hope I've convinced you of today is that airway remodeling is a consequence of asthma and unchecked inflammation. We need to know we need to know more about it, and we need to design drugs that will be able to effectively treat it so we can reduce this scar tissue formation. What I think, and what we're working on in my lab, is the role of MAP kinases as a therapeutic target. That means we want to inhibit it. And the way in which people around the world are inhibiting it with small molecule inhibitors, but what we're trying to do in my lab is to use something that's already there, an endogenous inhibitor, MKP1, and increase the amount of that, boost the amount of that. And what we're currently at is looking at post-translational modification of that, but we'll be refining that over the next couple of years as well. So, putting it all into one picture, if this is where inflammatory stimulation induces airway remodeling by this pathway and gene transcription, what we think is that MKP1 can block this and hopefully therefore have an effect on airway inflammation and remodeling. So I'd just like to end there by thanking you for your attention, very good attention and lovely answers to questions, and also to acknowledge, of course, research. I don't do the research all by myself. I have members of my team. This particular work was by a PhD student, Melanie Manich, who's from Switzerland, and Emma Ramsey, who's from Australia, and they've been working on this. These two guys, Freddie and Vera, are from Germany, and Anna Bjorkman is from Sweden, and he's from Australia. So we have a very international lab, and it's a very fun lab, and we hope that our research will be able to help human health in the future. So I thank you very much for your attention, and I ask for any questions.